हेलो फ्रेंड्स, दिस इज़ डॉक्टर रवि सैनी। आज जो हम टॉपिक डिस्कस करेंगे दैट इज़ ट्रांसपोर्ट ऑफ सब्सटेंसेस थ्रू द सेल मेम्ब्रेन। सो बेसिकली ड्यूरिंग various metabolic activity, either during catabolism or anabolism, various biomolecules transfers from one cell to another for specific biochemical reactions. So in this transport, how how does that transport occurs? What are the various concepts about it? Especially like in some of the transport, energy is used. In some of them, various mediator proteins are used. So all these concepts would be discussed in this chapter. Now come to the types of transport process. In this we have the first one is passive transport, in which we have a simple diffusion, facilitated diffusion, osmosis, filtration, bulk flow, solvent drag. And second one is the active transport, in which we have two categories. The first one is the primary active transport, second one is the secondary active transport. So basically इन दोनों के बीच में जो डिफरेंस होता है वो एनर्जी को लेके होता है. जो पैसिव ट्रांसपोर्ट से उनमें एनर्जी इज नॉट यूज्ड इन एटीपी फॉर्म, and in case of active transport, energy is used in ATP form. The third category is the vesicular transports, in which we have two subcategories, exocytosis and endocytosis. There is another fourth group, that is transport across epithelium. The first one is the by the mode of transcellular transport, and the second one is the paracellular transport. So सबसे पहला जो है that is a simple diffusion. So diffusion is a process of passive transport in which molecules move from the area of higher concentration to the area of lower concentration. So simple diffusion is the most simplest mode of transport, in which energy is not used definitely, and the movement of the molecules is from higher concentration to the lower concentration. In case of examples, basically जो gases का transport होता है, ऑक्सीजन, कार्बन डाइऑक्साइड, यूरिया और अमोनिया, वो इस मेथड के थ्रू होता है. Now come to the second, that is passive diffusion through protein channels or ion channels. So in this mode of transport, definitely energy is not used and it is passive, but there are some specific mediator protein channels or ion channels. So they are tubular structures. The protein channels or ion channels are tubular structures that connect the exterior with the interior of the cell. So basically इनका जो location है ion channels का और protein channels का, they are present in plasma membrane. So they connect the exterior of the cell to the interior of the cell. Now types of protein channels, we have water channels, we also call them aquaporins, and the second one are the ion channels. Now come to the ion channels. Ion channels are provided with gate on either side of the channel to control the movement of the molecules, and opening or closure of these gates regulates the movement of various ions through them. Basically there are three types of ion channels. The first one is the voltage gated, second one is ligand gated, and the third one is the mechanically gated channels. These channels have opening or closing gates, or by these opening or closing of these gates, they control the movement of various biomolecules. Voltage gated ion channels. Change in the membrane potential beyond a certain threshold value opens or closes the gate of these ion channels. Examples are sodium channels, potassium channels, and calcium channels. So जो सोडियम, पोटेशियम और कैल्शियम चैनल्स है, इनकी जो ओपनिंग और क्लोजिंग है, बेसिकली इट डिपेंड्स ऑन द डिफरेंस ऑफ मेम्ब्रेन पोटेंशियल. जैसे मेम्ब्रेन पोटेंशियल में कोई चेंज होगा, so this change would be responsible for opening and closing of these channels. Now the sodium channel. For sodium channels, the gate is located on the outer end of the channel. The gate closes the outer opening of the channel when the cell is at resting membrane potential, that is minus 70 millivolt in nerve. In case of muscles, it is minus 90 millivolt. and in case of pacemaker it is minus 50 millivolt. So basically जब cell resting stage पे है, so these gates are basically closed for these sodium channels. But when the inside becomes less negative, the gate opens and sodium influx occurs that leads to the first formation of local response and later the genesis of action potential, that is depolarization.